Hello dear students. Today we'll study the last segment of central dogma of flow of information, that is translation process. But before we come to translation, let's quickly recapitulate what are the different parts of central dogma which we have already studied. First is the structure of DNA. We have already studied the structure of DNA, and the most acceptable model is the double helical model proposed by Watson and Crick. We have studied it. Then this DNA, which is a blueprint of all the genetic information, has the ability to replicate, and the replication mode of replication is semi-conservative mode of replication. That was also explained by Watson and Crick. But experimentally it was proved by, yes, Michelson and Stahl through the experiment which we did, the heavy nitrogen N15 and N14. Do you remember? Yes. So, first step is DNA replication, through which it can make copy of the DNA. Then this DNA, it forms its transcript by the process of transcription, and it makes the mRNA, messenger RNA. There are cistrons which are basically coding for the proteins which are called structural genes. These cistrons are converted into mRNA, messenger RNA, and the information, that is the genetic information from DNA, is converted into messenger RNA. Now this messenger RNA has to be translated into proteins, and that process is called as translation. Today we will study this process of translation, which is very elaborate. So please, I am again stressing the whole process is very important, and any question can be asked for 5 marks. It may be the structure of DNA, the replication of DNA, then transcription process, structure of transcript, transcription unit, and then the most elaborate process is the translation. So today we will study this process of translation in detail. So this translation we can first divide into two parts. One, various requirements for this process, and then we will come to the various steps involved in the process. So let's see what all are required by the cell for the process of translation. So the first requirement is ribosomes, because ribosomes are known as the protein factories. On the surface of these ribosomes, the protein synthesis will take place. So these ribosomes, they are otherwise present in the form of subunits. Only during protein synthesis, only during translation, they associate. So let's see what all different types of ribosomes are required. So in prokaryotes, the types of ribosomes are 70S, and in eukaryotes they are 80S. S is the Swedberg's unit or sedimentation coefficient. This 70S is actually made up of 50S and 30S subunits. Only during translation these 50S and 30S will come together. Likewise, in eukaryotes, the ribosomes are 80S, which are made up of 60S and 40S subunits. Fine. So the first requirement are the ribosomes. Then mRNA. These subunits of ribosomes will associate only when mRNA is released into the cytoplasm. So second is mRNA. And this mRNA also in case of prokaryotes, The mRNA, as soon as it is produced, it can go for translation because no processing is required for the mRNA in prokaryotes. But in case of eukaryotes, after the RNA is produced, first it is called as hnRNA, that is heterogeneous nuclear RNA, and it has got certain areas which are called as exons and introns. Exons are required for translation, whereas introns have to be removed. Plus, there are other two processes. One is capping and other is tailing. So in all there are three steps in processing: capping, splicing and tailing. So then only the hnRNA, heterogeneous nuclear RNA, is converted into mRNA in eukaryotes. In prokaryotes, it's not there. Next is tRNA. In last class, I explained you the structure of tRNA. tRNA is a very big molecule. It's a folded molecule and it gives an appearance of clover leaf-like structure. It is also called adapter because of its arms, it behaves like an eye as well as hand. You know that there are four ends. First is the amino acid end. Opposite to amino acid end, there is anticodon loop. That anticodon loop acts like an eye, and that amino acid end acts like the hand. So based on the anticodon, a specific type of amino acid is picked up by the tRNA. We'll study it in detail. Then there are different amino acids which are present in the cell pool of the cell. So they are required because protein synthesis requires amino acids, because proteins are made up of the amino acids by peptide bond formation. So amino acids are also required. Then the very important enzyme which is required for translation process is amino acyl tRNA synthetase. This is a very important enzyme which is required for charging the amino acids as well as tRNA. Then we require energy currency, ATP, also GTP, and there are various protein factors required: initiation factor, elongation factor and termination factors. Various different types of factors are required. So these are the requirements for the process of translation. Now let's switch over to the steps which are involved in translation process. First step is activation of amino acid, because there are so many different types of amino acids present in the cell pool. The specific type of amino acid has to be first activated based on the information present on the mRNA. Then this amino acid has to be transferred to tRNA, and then the process begins. The first step after transfer of amino acid to tRNA is initiation. So initiation of polypeptide chain begins, then elongation of polypeptide chain is there, and finally there is termination of polypeptide chain. And after termination, whatever the polypeptide is produced, that undergoes post-translation processing to form a specific type of protein. We know that proteins may be of different types. It may be primary, secondary, tertiary, quaternary. So based on the requirement, there are specific types of modifications which are taking place after translation. Okay, so these are the various steps for the translation process. Now let's study all this process through various diagrams. Now let's study the whole process of translation with the help of the diagrams. You know, the first step is the activation of amino acid. So first the amino acids, they are activated by the enzyme. You know the enzyme? Enzyme is amino acyl tRNA synthetase. Along with the tRNA, in presence of ATP, there is the charging of the amino acid with the tRNA. Okay, so this amino acid and tRNA, they get binded. So the amino acid will get binded here based on the anticodon present here. For example, if it is UAC anticodon, so the amino acid for AUG will be activated, which is methionine. You know, methionine is a dual purpose amino codon. It is a start codon as well as it codes for amino acid methionine. Okay, so AUG is a dual genetic code. So the first amino acid will always be the AUG, that is methionine. So first the amino acid methionine will be activated and attached to the tRNA. There will be the use of ATP in presence of the enzyme amino acyl tRNA synthetase, and then amino acyl tRNA complex will be formed. This is amino acyl tRNA complex, and then AMP is released. Okay, so the first step is charging of amino acids. Now, as soon as the amino acids are charged, these subunits of ribosomes, suppose we are talking about a prokaryote, it will be 50S and this will be 30S. So this will be 50S and this will be 30S. These two subunits will associate and they will form certain imaginary groups which are called as A site and P site. A stands for amino acid site and P stands for peptide site. Okay, so this is peptide site. A new peptide bond formation will take place here and a fresh amino acid will come and bind to this place. Okay, so there will be two sites. One is the peptidal sites, P site, and A is the amino acid site. So they associate together. Then as soon as they associate, the first amino acid which is brought because of this genetic information. See, this is mRNA. This one is mRNA. This mRNA is always read from 5' to 3'. So the first code at the 5' end is AUG, that is a start code, start codon. So AUG is a start codon as well as the codon for methionine. So this tRNA having anticodon UAC will be charged and it will bring this methionine. The first amino acid, that is methionine, is always brought to this position only. So the first is methionine. Now second amino acid is UUU. The genetic code is UUU. UUU is coded for phenylalanine having the anticodon AAA. So now the next tRNA having phenylalanine will come here. This is the initiation. For this process, initiation factors are required. There are three initiation factors, IF1, IF2, IF3 are required for the prokaryotic cell. Whereas in case of eukaryotes, there are 9 IF factors required. We'll not go into the details of it. It's not required, but you must know initiation factors are required. So this is the initiation factor. So initiation of the polypeptide chain begins. Now, still it's just the initiation. Polypeptide bond formation has not yet begun. Now what will happen? This mRNA will move this way, or you can say first amino acid, that is methionine, it will come at the P site. Second amino acid will come at the A site. Now this sequence has moved forward. So what will happen? This portion will go here and this portion will move here. So let's see what happens. So now the chain has shifted here. We know because the chain has shifted here, this chain has got phenylalanine and this methionine will go and it will form a peptide bond here. So a peptide bond formation takes place between the methionine and phenylalanine. Now it has come to the P site. So the peptide bond formation will take place only at the P site. Now since the sequence has moved forward, the sequence which was there after UUU will come to the A site now. Now this AAA, this is coded by, this is coding for lysine. Okay. So this lysine will come here. Now the new amino acid, the tRNA having amino acid lysine will come and bind here. Now initiation of polypeptide formation takes place. Now it will be the elongation. Okay, this process you must know. Changing of position from A site to P site is called translocation. When the tRNA, here this tRNA is at the A site. When this mRNA moves forward, the position is changed from A site to P site. This process is called as translocation. So you must know the difference. Sometimes you may be asked to differentiate between translation and translocation. Translocation is just a step in translation process. It's just the position, the change of the position of the tRNA from A site to P site. That is called translocation. Now, this place is occupied by lysine. Again, what will happen? The chain will move forward. Now, this position will come to P site. So, what will happen? Now, this tRNA having lysine will come here, whereas this will break up and this will form the polypeptide. Okay. Now, the tRNA which is attached to methionine is removed, disassociates. Also, the tRNA which is attached to the phenylalanine also has disassociated and peptide bond formation takes place. So this is called elongation of the polypeptide. In this way, depending upon whatever sequence is there in the mRNA, this polypeptide bond formation will keep on continuing. This is called elongation of polypeptide chain. Now you can see after AAA, it is the sequence is GGG. GGG is actually the code for glycine. So the tRNA will bring lysine having anticodon CCC will be activated and the glycine will be brought here. So this glycine will be brought having CCC. So now this glycine will come to the site of this part, this A site. Okay, again the translocation process takes place. Now again this will move forward and the peptide bond formation will be there. So let's see how it will take place. So here it is glycine, next is lysine, next is phenylalanine, and then it is methionine. So this way the elongation will keep on increasing by peptide bond formation. Okay, so the elongation will continue till the termination codon comes. Now you can see at A position the codon which has come is UAG. Does UAG code for any amino acid? No, because UAG is a stop codon. There is no amino acid coded by UAG. That's why it is called as a stop codon or termination codon. So since there is no amino acid for UAG, so no fresh amino acid will come here, and this will give a signal to the whole process that now it's the time to terminate the process of this polypeptide chain formation. Okay, so the termination factors will operate and they will disassociate the ribosomes, and this chain of polypeptide amino acids, that is polypeptide, will be released. Okay, so as a result what will happen? A chain having methionine, phenylalanine, lysine, glycine, this will be released as a polypeptide. Okay, and with the help of the termination factor and releasing factor, this polypeptide is released. So this process ends, and these ribosomal subunits, they will again disassociate. Okay, and you must remember, so on the same chain many ribosomes can associate. Likewise here, so what happens at a time, many ribosomes may produce the polypeptide chain on the same mRNA, and as soon as the UAG comes or termination codon comes, these subunits will separate and the process will be terminated. Okay, so this is the complete process of translation, and once this polypeptide chain is formed, this undergoes the post-translation processing. Depending upon the requirement, there are further, you can say, modifications are done. So primary protein, secondary protein, tertiary protein, whatever is required, that modification is done. So this is the whole process of the translation process. Okay, there are certain segments which, though they are present, but they do not undergo translation. They are called UTR, untranslated regions of the mRNA, which are not, they are present on the transcription unit, present on the mRNA, but at times they are not showing the translation process. But these are helping in the process of the translation. Okay, so with this we have completed the whole process of translation. Whatever steps are required along with the diagram, you must practice. This question may be asked for 5 marks. I hope you have understood the whole process of central dogma of flow of genetic information. Thank you.